Hello, good morning.
Good morning.
You can hear me?
Yes, we can.
Okay. Okay, fine. So we're gonna start at this moment. So we are going to study the immunity to infections, and specifically we'll be looking at the immunity to bacterial and viral infections. The learning objectives that we should be able to discuss through these few minutes. You need to understand the following: that is the root and sites of infection, mechanisms of tissue injury regarding infection, the timing of the immune response to infection. Remember, there is always the acute, and later you have the chronic infections. Then you also need to understand how the immune system regulates cell-mediated immune responses, and that is by looking at the humoral immunity versus the cell-mediated immune responses, and the effector mechanisms that are required to implement these immune responses. So the key determinant is to look at how these different types of antigens trigger different type of immune responses. We also have the microfill evasion of the immune responses, and these are evasive strategies that some of the pathogens have evolved so that they can overcome the host immune response. Now, just in a quick overview, let's have these questions in mind as we discuss. What is immunity? I believe you know what it is, the defense mechanism that exists at any given time, and that is what is available to deal with any availability of infection or injury for that reason. Now, what is the infectious dose? Remember, there are certain number, or rather the magnitude of infections or infectious agents that should be able to trigger an infection. If it is so much less, you understand that this can be dealt with easily, but if it becomes more than it can be withheld or rather controlled by the immune system, then chances are that the immune system will be overwhelmed by this number of pathogens at any given time. So the dose is very important. You need to figure out that. You also need to figure out that do all infections cause harm or injury to the host once the infection has been established? How does the immune system respond to different pathogens? I've said this one earlier on. You know, we have viruses, we have bacteria, we have allergens. All these are different antigens. But when we talk about infectious agents, then we talk about the transmissible diseases that require an agent. It could be a virus, a protozoa, bacteria, and all that. But I'm also bringing the issue of allergens in the context that they are not infectious agents. The immune system should recognize this as an allergen as well. So you also need to understand what happens when an immune system fails to prevent or rather eliminate an infection, and what are the strategies that are employed by pathogens to evade the host immune responses. And lastly, just to maybe bring this on board, the role of vaccines, because they are very important in boosting the immune response whenever there is an infection, either within the immune system or preventing for a similar occurrence in future. Now, what are some of the steps that are needed to establish infection? Now, this is what you already, I think, must have gone through: the innate immune mechanisms and adaptive immune mechanisms that are very key in dealing with the immune responses or the immunity against certain pathogens. So these steps simply require the following to happen: that the pathogen should be able to penetrate the physical barriers or the mechanical barriers, in this case, the skin, the gastrointestinal lining, the respiratory lining, and also there needs to be competition with the normal flora for binding sites. Remember, you have these different normal flora within the body services, like the lactobacillus acidophilus in the vagina, there is the bifidobacterium bifidus, and also, of course, there are other um, pathogens, or rather, not pathogens, but rather the normal flora, which can translate into pathogens whenever there is an injury. So by having this colonization of the body surfaces by certain normal flora, it doesn't guarantee you that they will be continue to be a normal flora. Later on, should there be injury, the same normal flora can cause disease. Of course, then you need to understand the invasive nature of the pathogens, and this has to do with evading the innate immune mechanism, which is comprises of the macrophages, neutrophils, and natural killer cells. These are the very key cells from the innate immunity that fight against infection. Now, the diagram just tries to emphasize that for the immune response to reach the site of infection, certain things must happen under the process of inflammatory response. And that is bringing us to this question: how do phagocytes invade the area of infection or injury? As part of inflammatory response, certain factors are released. For example, there is the histamines are released by the mast cells, and this has a lot to play with vasodilation, that's stimulating the capillaries become more permeable, and this leads to the next stage, that is imagination. Remember, the site of infection, the blood vessels, including capillaries, they expand in diameter, now making the blood to sit down, and this blood has with it the immune cells, the white blood cells, which now must migrate from the central area of circulation to the periphery of the capillary or the blood vessels, and they need to move out of that capillary into the tissue itself. Now that is the process of diabetes, where the blood cells leave the vessels and enter the site of infection. And then there is also the attraction, because certain protein molecules are also activated, like the complement system, which sometimes has some capability to attract the immune cells to the site of infection. So this chemotaxis, you can learn it maybe later on as part of the complement strategy to attract the immune cells whenever it has been activated by the presence of antigen-antibody interactions. Now, the roots of infection, like I said earlier on, the infection cannot happen unless there is a mere entry or the breaking of the physical barriers, which now will trigger the immune system to start acting against the pathogen. So first stage is to have the adherence to the epithelium of the skin surface by the pathogen. And this, if there is nothing that happens or the barrier is not broken, then that infection can be protected. Sometimes the skin has the dead cells, which also creates a major barrier towards this infection. However, sometimes due to other injury, like for example, the physical injury, you might find this physical barrier is being compromised. And that leads to entry or inhibition of these pathogens to the next level of the skin. And as the diagram clearly indicates, that the pathogen now is able to enter the skin and even to the lower or the further inside part of the skin. And within there, the physical barriers includes the skin, the chemical molecules like the pH, lysosomes, which are part of the saliva. All these are just very key in terms of preventing the entry of the bacteria and also killing the pathogens. But should that not happen, then you'll have that there's entry of pathogens, or rather there's infection that has taken place, and this has to deal with the second line of defense, which comprises of the innate immunity. Now, the innate immunity comprises of these innate immune cells, which we have discussed earlier on, the macrophages, the neutrophils, the dendritic cells, and the eosinophils, the neutrophils, and these are very, very, very key immune cells, as you might have studied them earlier on. So should this second line of immune defense, that is the innate immune cells, once they have been recruited, they can deal with the infection, but if they don't completely eliminate some of them, like the macrophages, they are also antigen presenting cells. And at that time that they have already phagocytosed or endocytosed this pathogen, they can produce other proteins within their cell, that is the MHC molecules, which can bind with these broken antigens, and this can be presented for T-cell recognition, and that attracts the specific arm of immune responses. Just before I leave this page, I want to reiterate the fact that the normal flora and local chemical factors are very key in inhibiting microbial growth at the surfaces of certain body areas, and majorly these are the skin. But I said earlier on that should there be a compromised state like immunodeficiency, some of these normal flora sometimes can cause disease if they get access to the host or the internal cells. Then the wound healing sometimes is induced by antimicrobial proteins, and these are the fatty acids that are actually expressed at the skin level, and they are very important in killing bacteria. But in addition to this, other soluble molecules like the complement, these are special proteins that are released from the liver into the blood circulation. They can only be activated once there is an antigen antibody interaction, and this complement can also play part in enhancing the immune responses by its activated complement factors and the protein molecules that you must have learned under the complement system. But nevertheless, the complement also behaves like antibodies in the sense that they can interact with antigen antibody complexes and expedite that process called opsonization, which enhances killing by the phagocytic cells. Now, the innate immunity cells express receptors, and these receptors are very key in recognizing pathogens. As you all know that these PAMs, or the pathogen-associated molecular patterns, you also find them on the outside of the pathogens. And these are the ones that interact with pattern recognition receptors, which are found on the immune cells. So the pattern recognition receptors from the immune cells should be able to recognize the PAMs that are found on the pathogens. And this interaction causes activation of target immune cells. Now, these pattern recognition receptors, we have examples as toll-like receptors, which recognizes lipopolysaccharides that you find on gram-negative bacteria. We have also toll-like receptor 2, which recognizes beta-glycan. These are proteins that are found on gram-positive bacteria. Then you also have the toll-like receptor 3, 7, and 9. And these are specifically important in recognizing the viral nucleic acids. So these toll-like receptors are actually pattern recognition receptors, which are expressed on the surface of the innate immune cells, and they can be able to recognize these different types of pathogens. So get to note that each receptor is specific for certain antigen or pathogen. Now, viruses, in addition, once they have been introduced, or once there is infection that has taken place, they can trigger the immune cells to produce special proteins which we call interferons. And these interferons have been divided into three categories. There is interferon gamma, alpha, beta, and these are very crucial, especially in producing antiviral state, meaning that they act on a neighborhood cell or on a nearby cell, and this cell is able to protect itself from invasion by this particular virus. So interferons have been used for many years as part of the treatment for viral infections, which actually is the current practice even with hepatitis C and hepatitis B viral infections. Now, the root of infection can be extended right from the innate immune response to adaptive immune response, in the sense that once you have the infection that has established, this infection has to be dealt with by both arms of the immune system. So I have said earlier on, like the part of the innate immune cells, like the dendritic cells, the macrophages, they are capable of phagocytosing this particular antigen, presenting it with the MHC molecules that are expressed on their cell, and this, once coupled with an antigen, depending, because these are MHC class 2 molecules, they will be recognized by the T helper cells. Now, in that way, the T helper cells can activate more other cells from the innate system, as well as they can also activate the B cells to produce antibodies. Now, if you see the diagram, it gives you that from the tissues, you have tissues which have also been infested by pathogens. Within the tissues, you have cells. Now, cells sometimes have some spaces within them, interstitial spaces, which sometimes are enriched with lymph. Now, this lymph is just a fluid, a colorless fluid that can also play a critical role in transporting the pathogen to lymphatic vessels where the lymph circulates, and this sometimes can take them through the lymph nodes, which are very rich in immune cells, and this one can act as gates to filter out infection. So in that way, you find like there is involvement of both innate immune response and adaptive immune response, whereby in specific or in adaptive immune response, you have the B cells being activated, and that's what you can see here is that the antibodies are being formed, which can also bind with the antigens and either block or neutralize the infection for that reason. So we can now look at the infection compartments. As we have looked previously, that you always start with the surface where there is physical barrier. But should there be a compromise in the surface, then obviously you have an infection that has taken place, the near entry of a pathogen under the immune system. And these cells of the immune system are very, very different in terms of which type of antigen they can handle. Now, the macrophages are the most popular phagocytic cells from the innate immune system, and this has evolved with many receptors to deal with different pathogens ranging from viruses to bacteria and so on. And now what happens here is that within the interstitial spaces, and these are the spaces between cells, if you look at the liver, if you look at the kidney, they have similar cells, but from one cell to the other, there is a space in the middle or between them, and this is what we call the interstitial space, which can be occupied by either blood or lymph. And within this site of infection, you can also have different pathogens infecting that particular area. You can have virus, bacteria, protozoa, fungi, and all that. Then remember viruses are also intracellular pathogens. So as we look at these infection compartments, bear in mind that certain pathogens can only exhibit themselves as extracellular, meaning they can only stay outside the cell. And then we have seen examples of interstitial spaces and also these epithelial surfaces. These are just, yes, it can be inside the body, but not inside the cell. So these pathogens can still be residing within those spaces or those areas that we have mentioned. Now we have also the intracellular pathogens, and these are the pathogens that enter the cell, the target cell, and establish itself. It can replicate inside that and produce those foreign proteins which can be recognized by the immune cells, especially depending if they are presented with MHC class 1 or MHC class 2. Because if you remember your body cells, they have MHC class 1. Only the antigen presenting cells have MHC class 2. So while the macrophage, for example, can be critical in presenting antigens, but it can also be infected. And now, being an antigen presenting cells, it has MHC class 2. So in that way, this pathogen, or I mean, this antigen presenting cell can also be infected. So those pathogens like Mycobacterium tuberculosis, they can also infect the macrophages, and that's why we can talk about certain pathogens being intracellular, while the others can also be extracellular. So in such a case, we talk about different pathogens that can infect either extracellularly or intracellularly, and examples are given there. I'm not going to go through each, but bear in mind these examples because whether it is intracellular or extracellular, the infection has already taken place and it has to be dealt with. Now, the protective immunity against extracellular pathogens as well as intracellular pathogens is different. For example, antibodies are a very good immune product that can deal with extracellular pathogens by different mechanisms, either by neutralization or by opsonization. And this we are going to see shortly. Then in terms of intracellular pathogens, the antibodies will not be effective because they cannot enter inside the cell. And in that case, we have the natural killer cells as well as the cytotoxic T cells, which are very, very important in dealing with intracellular pathogens. Now, how do they do it? I said earlier on, like when you have infection within the cell, the expression of foreign proteins have to be recognized by that particular cell that is infected. And this has to be coupled with an MHC molecule. Now, for the sake of cytotoxic T cells, they only recognize an antigen that has been processed and coupled with an MHC molecule. That becomes a target by the CD, the T cells. Now, depending on which type of MHC class that is sold, if it's MHC class 1, that becomes a target by cytotoxic T cells. An example, for example, if you have hepatitis cell or a liver cell that is infected by hepatitis virus, that liver cell has MHC class 1, and that becomes a target by the cytotoxic T cell. If, for example, you have the natural killer cell that doesn't require an MHC antigen complex, but rather it has receptors on its surface, that is the natural killer activating receptors and inhibitory receptors. And it does the monitoring of MHC expression on the infected cell. So cells that have been infected by the viruses, usually they have minimal or reduced MHC expression. And that becomes a target by the natural killer cells. So whenever there is a minimized or reduced MHC expression, the natural killer cells, through its natural killer activating receptor, it is able to recognize that this particular cell is infected, and it has to deal with it by releasing the granzymes and the buffering, and these are lytic molecules which is able to cause lysis on the target cell, which is the same molecules that are produced by the T cells in killing the target cell or in lysing the target cell. Now, the mechanisms of tissue injury during infection. Now, sometimes you may wonder, why is it important that you need to get worried about these infections, either by viral infection or by bacterial infection and any other pathogen? The reason is we have different target tissues or cells. Some of them are a bit critical because of the nature of their work they do in the in the organism. And you can imagine if, for example, a shoulder muscle, even if it's infected, it may not be that critical when you compare with the heart muscle or during endocarditis when the heart muscle is inflamed. And you know the work of the heart is just to pump. If the muscles are infected, then the heart will be compromised in terms of its capacity. So, and that is why we have to look at this direct mechanism of tissue damage by pathogens. Now, let's have a look at the examples that are given here. We have direct mechanisms of tissue injury by pathogens. For example, we have the exotoxin production. And these are the toxins that are produced by pathogens. And the examples are produced by the Staphylococcus pyogenes, Staphylococcus aureus, and the group, as you can see. These are the pathogens that release the toxins. And these toxins are released outside the cell or the pathogen. And this one can cause hypersensitivity reaction. In other words, there is exaggerated immune response. And whenever these responses are being produced, they even injure the neighborhood or the healthy cells. And for that reason, it causes a problem. Sometimes there can be overstimulation of the immune system to produce more cytokines by overactivation of the T cells. And this is some of the damage that is very critical even than the pathogen itself. Now, the other product that is produced is the endotoxins, which are part of the cell membrane of gram-negative bacteria. And this can only be released when this bacteria or the pathogen is lysed. And for that reason, they can also cause the same problem of inflammatory response, just like the exotoxins. And the examples of those pathogens that produce endotoxins have been given there is Escherichia coli, Anopheles influenza, Salmonella type, Shigella, Pseudomonas, and so forth. Now, sometimes these presence of these pathogens can cause certain physical changes within the cell that they have infected, what we call as direct cytopathic effect. And this has to do with the morphological changes. So they can make the cells either to become larger, to be swollen. And these examples have been given there, mostly the viral infections. And you can imagine these are some of the complications that can happen when you have the critical cell that is infected, especially like the heart or the nervous system and so forth. So this can have far-reaching implications in terms of the functionality of that organism. Now, the diseases that are associated with this exotoxin, endotoxin, have been given there. And part of this is the inflammatory response or the inflammation that is caused by this. And that's why I was even saying earlier on that if you have, for example, the inflammation of the meninges, that is the covering of the brain, and you have meningitis, you can imagine the disaster that will happen. So some of these pathogens need to be eliminated because of that activated immune response that will happen, and it can be life-threatening to the individual. So these are some of the implications of the mechanisms of damage because of the release of the toxins, whether it's endotoxin and exotoxin. And remember, these toxins, whether it's endotoxin or exotoxins, they are what we refer as to superantigens, meaning they can activate the T cells without necessarily having to go the process of antigen presentation, just like the normal way that antigens are recognized by the innate immune system, presented for T cell recognition. And finally, the T cell generates immune response. But with the super antigens, includes now these exotoxins and endotoxins, they can activate the T cells by attaching to excessive secretion of cytokines, which can cause a lot of inflammatory response with far-reaching implication like toxic shock syndrome, which can happen because of excessive activation of the T cells, and that can be life-threatening. Sometimes it can lead to death. So this has to be controlled. Now, there is also the indirect mechanism that are caused by damage, tissue damage by these pathogens. And this is usually by the formation of immune complexes, because this deposition of immune complexes sometimes attracts the continuous presence of immune cells in trying to deal with that immune complex. And sometimes you have these chronic diseases like arthritis, where you find the immune complex deposition at the joints, causing this arthritis. You have also the anti-host antibody. Sometimes when you have the release of these antibodies sometimes can react towards the body cell antigens and cause what we call autoimmune disease. For example, in streptococcal infection, like the streptococcal that you can have, or that can overstay for some time, it can lead to the damage of heart muscle, simply because sometimes when there's this presence of an antigen, antibodies can be produced. But sometimes these antibodies can recognize the heart muscle as an antigen without necessarily differentiating between the real antigen and the heart muscle, because they have what we call molecular mimicry, where the antigen, the self-antigen can share some similarities with the antigen from the pathogen itself. And in that way, the antibody that is released can bind and attract the autoimmune disease for that reason. We also have the cell-mediated immunity, where we find, like in case of Mycobacterium tuberculosis and others, where you have chronic infection, meaning like in the lungs, you have the presence of Mycobacterium tuberculosis, which attracts the macrophages. Now, the macrophages come in an event that they want to make a wall to prevent further spread. And now, when you have that continuous recruitment of macrophages to the site of infection, you have a development of granuloma, which is a classical example that can be even examined under a microscope as the manifestation of TB. And this causes hypersensitivity reaction, what we call delayed type hypersensitivity, where you have continuous presence of recruitment of immune cells, because the pathogen itself also continuously infects other immune cells by using its evasive strategies. And that causes chronic inflammatory response with far-reaching implications that sometimes it can lead to death. So these are some of the indirect mechanisms that you can have tissue damage, which you can differentiate from the direct. From the direct, you have the release of toxins, but the indirect is the formation of these antibody antigen complexes, which can continuously attract the presence of the immune response. For example, in the liver disease, where you have the hepatitis viral infection, the major damage or the necrosis that happens is not due to the bacteria, not due to the virus itself, but rather the presence of the release of the granzymes or the enzyme, the bufferings by the cytotoxic T cells, which now causes the necrosis of the liver cells. And the many examples are given there. Now, in terms of timing of the immune response, as you all know that you have the initial infection that can last for few hours. If it's not dealt with, it can expand to 2 days. And then you can have it for longer than 48 hours. Now that it goes into more of chronic state. So there is the initial cells, you have the phagocytic cells, then you have the recruitment of the other specific arm of immunity by releasing certain cytokines. And finally, you have the immunological memory if you have generation of antibodies or plasma cells, or you have T memory cells that have been generated. So when we talk about the timing, it's the time that it takes for establishment of immunological memory and whether this pathogen will be ever cleared right from the innate immune system, or it has to be persistent, like in the case when the pathogen evolves or develops evasive strategies against the immune response. And usually we have this as acute phase and the chronic phase, depending on how the immune system works with that particular antigen. And at this point in time, remember, we are different in terms of the immunity that we have at any given time. So we may not respond the same to the presence of same antigen. Now, in terms of mucosal immunity, we have the polymeric immunoglobulin A, which is associated with mucosal immunity. It usually transported into the gut lumen through the epithelial cell surfaces at the base of the cubes. And when you see what is happening is that this immunoglobulin A finally will reach the target antigen. It could later be a bacterium or its toxin, and it will bind to that particular toxin. Now that will render it less injurious to the body system because it's probably neutralized or blocked if it's an antigen like a pathogen from attaching to another cell. Now, the diagram is just showing you how the macrophage is very critical in terms of linking between innate and specific arm of the immune response. As much as it is from innate, it has evolved to have different receptors that can recognize different antigens from the pathogens. We have receptor for mannose, we have receptor for glucan, we have receptors for many other antigens that you find on the surface of antigens. And these are very important for recognition. If you have no recognition, then no immune response can take place. So the bacteria binding to the macrophage receptors initiate the release of cytokines, and this usually is interferon gamma, which can attract or activate the T cells and activate autoimmune cells from the innate system, and this will kind of amplify more response to the same pathogen should there be continuous replication of the same pathogen. Now, the macrophages, on the other hand, they are also phagocytic cells, meaning they can ingest, that is engulf and ingest the bacteria to which they bind. And they have what we call lysosome. This is a bag of lytic enzymes or a vesicle that is rich in lytic enzymes, and we call them lysozymes. And these can be released to break down the ingested pathogen when it fuses with the phagosome, and when they make what we call phagolysosome, which is now the last stage where this antigen has to be broken down and presented with MHC molecules. So MHC molecules are expressed once this antigen has been ingested, and it's able to bind with MHC molecule, and that is now the target for T cell recognition. On the other hand, what this gives you is the T helper cell, which is very critical in activating the B cells to produce antibodies. Like the diagram shows you is that the T cell has this role of helping other cells to function. So they can activate B cells to produce antibodies, and these antibodies can bring about neutralization of an antigen by binding with the epitopes and blocking its attachment to the cell, or these antibodies can bind with the antigen, and that one can make it more of or enhance phagocytic activity whenever the antibody has binded with an antigen, and this attracts the phagocytic cells, and this process we call it opsonization. Or this antibody can also bind whenever there is an antigen, and this will also attract the complement, which is another protein that is able to assist the clearance of pathogens from the body itself. So whenever there is an antibody antigen complex, the complement will come and bind with that antigen antibody complex, and this also enhances the phagocytic process and the elimination of that particular pathogen. Now, this diagram, you don't have to worry why I just brought it just to make you understand the role of the complement. The first part is that the complement is very critical because it binds with an antigen that is a pathogen, and that one expedited the process of killing. As you can see, the macrophage is able to attract that or get attracted to that binding of a complement. It's also very critical in inflammation. I said earlier on that the complement involves a cascade of proteins that are activated in a particular way so that one activates each other. And at the end of the day, there is cleavage of certain proteins into the A molecule and the B molecule. So the B molecules are very important in opsonization, while the A component, like the C3A and the C5A, they are very important in inflammatory response, meaning they are chemoattractants of the immune cells. And also the complement sometimes can bind to certain molecules on the surface of the pathogens and cause lysis, what we call the membrane attack complex. And this one is very, very critical in killing or lysing the bacteria. And also the complement sometimes binds with antigen antibody complexes, and this makes it possible to eliminate some complexes via blood, which can happen at any time in the body. So these are some of the processes by which the complement brings about elimination of pathogens from the body itself. So that is clear now. I think we can look at this table, which is trying to re-emphasize what I said in terms of the action of antibodies. Antibodies can bind with toxins, and these toxins sometimes can help in preventing any infection, and that is neutralization, because the free binding sites are no longer available. And I've said this, antibodies sometimes also can bind with antigens and enhance phagocytosis. And lastly, I've said it can also attract the complement, and causes lysis and ingestion by the phagocytic cells. It's just the same thing that we repeated for the sake of clarity and all that. And this gives you now the mechanism by which the T cells work. The first one is the cytotoxic T cell, which recognizes any antigen in association with MHC class 1, and this becomes a target by the CD8. And as you can see, finally this target cell will be lysed. In which way? The CD8 cells release perforin and granzyme, which pokes or rather makes holes on the target cell and causes lysis, because there's an influx of calcium, ions, and the water, and that causes the lysis of the cell. While the T cells have these two categories, we have T helper type 1, which activates the intracellular killing, especially by making sure that this phagocytic cell is, like for example, a macrophage, is able to express the lysosome. We say the lysosome is just a bag of lytic enzymes. We should be able to break down that ingested bacteria and make it into small fragments. Sometimes these are the same fragment that can be coupled with an MHC molecule, which also gets synthesized as this macrophage has already ingested that particular pathogen. So that expression of MHC can bind with an antigen and present this for the T helper cell recognition. But sometimes this pathogen can be killed by those lysosomes, and then the only thing is that this pathogen can only the broken components can only be coupled with MHC and recognized, presented for the T cell recognition for future protection by activating B cells to produce antibodies that can recognize the same antigen. While the T helper type 2 cells are also very important because they activate the B cells to produce antibodies. So depending on the nature of antigen, you can either have the T helper type 1 being involved or the T helper type 2. So for the sake of clarity, the T helper type 2 is basically important in activating the production of antibody by activating B cells, while the T helper type 1 usually activates the immune cells like the macrophages to also activate other innate immune cells or macrophages by releasing cytokines that attract each other. So it is very critical that way that you understand the slight difference between T helper type 1 and T helper type 2. And remember, whenever you have also other antigens, not necessarily pathogens, for example, the allergens, the T helper type 2 can preferentially activate B cells to produce one specific antibody, and that is immunoglobulin E, which will bind with that particular antigen, and it causes it to be eliminated or rather to be neutralized by that immunoglobulin E specific antibody. Now the diagram may look a bit complex, but it is quite simple. It's just to bring you the difference between T helper type 1 and the T helper type 2. If you can see, both of them are activated by the mere presentation of an antigen by an antigen presenting cell. And then what happens is the difference in terms of which cytokines are released in the end. If you see type 1, you have interferon gamma, tumor necrosis factor, beta, and interleukin 2 as the major cytokines that are produced by T helper type 1. While T helper type 2, it releases interleukin 4, interleukin 5, interleukin 10, and interleukin 13. That is what I want you to get to know regarding this table. You don't have to understand everything in it, but get to know that the T helper cells, mainly type 1, is able to generate this particular cytokines, where the T helper type 2 also generate this particular cytokines. Now, immune response to viral infections. I'm now trying to end up what I've been talking. That first step is to deal with the innate immune cells, and this is a general response to any infection. It's not only restricted to a viral infection, but even bacterial infection. The immune cells, like we have said, they have the PAMs, the PRR, which interacts with the PAMs of the bacteria or the viral infections, and this is very critical. So by utilizing the pumps from the pathogen side and the PRR, that's pattern recognition receptors from the immune cells, then this pathogen can be recognized, interacted with by the innate immune cell, and presented. In this case, we are talking about the viral infections. So the molecular patterns that are found on the virus will be recognized by the innate immune cells through their pattern recognition receptors. Now, when a cell is infected by a virus, this cell will release interferons, and we have said it before. The interferons will not act on the same cell, but rather on the nearby cell. And now, this we are going to study, look at briefly what happens. So the immune response against the virus is primarily through interferons, and we have this double-stranded RNA, which induces the production of interferon gamma. We have also the main producers of the interferon gamma and interferon beta, which are what? Dendritic cells. And then no wonder, like in case of mucosal infections, we have the availability of dendritic cells, which are very key in mucosal services. So they can be able to produce these interferons as one way of trying to prevent further spread of these infections. So they are the ones sometimes getting infected. I think in case of HIV infection, we have seen studies that are looking at the role of dendritic cells in HIV infection. Now we have the toll-like receptor 3, which is pattern recognition receptor that recognizes the double-stranded RNA, and also the toll-like receptor, which is a pattern recognition receptor that recognizes single-stranded RNA molecules. And these are just the genetic material that the virus have whenever they have infected the target cell. So these interferons can produce an antiviral state by inhibiting the viral replication and at the same time inhibiting the viral infection. How does this one happen? We can see in this table. It looks a little bit complex, but get it to understand that each cell will have a receptor for these interferon molecules. Now, once this receptor recognizes this interferon gamma, what happens? A cascade of events happens within that particular cell. And this is a healthy cell that has just interacted with interferon molecule. And finally, certain protein kinases will be activated by the attachment of this interferon gamma on the receptors on the cell surface. And finally, what will be produced is the antiviral proteins, which can be able to break down this viral RNA that is being made and maybe make it degenerate, or sometimes that prevents the viral replication, because some of these proteins are required for viral replication. And this explanation is given there that there's an expression of the 2,5' oligoadenylate synthesis, which is an enzyme that is very critical in degrading the messenger RNA that has been formed in that particular cell. In addition, there is also the activation of RNA-dependent protein kinase, which is achieved through phosphorylation of eukaryotic initiation factor 2. There's also the protein synthesis that is inhibited, and this also causes the viral replication not to continue. So there is a lot that goes than what I've just said within the cell, but what is very key here is that the viral replication is stopped for further transmission to take place. Now, the interferon signaling is just a summary of what I've said. You have the first cell that is infected. It releases this interferon. Now, this interferon can also activate the normal cell down there to synthesize these antiviral proteins. And whenever these proteins are produced, they can also be a very good messengers to activate other innate immune cells, like the natural killer cells, to produce other cytokines which can activate cells, and also, or they can also be activating this natural killer cell to produce the buffering and the granzymes, and this can target the infected cells and cause that cell to be lynched or to be killed. So these are some of the ways that those interferons work. They can work by either attaching to a new cell or a nearby cell, or sometimes they can also activate other innate immune cells to also cascade the preventive mechanisms, and also they can activate other innate immune cells like macrophages to target those infected cells. Now, the step 2 of infection involves dealing with viral infection involves adaptive immune response, and that is the specific arm of immunity which requires the T cell and the B cell involvement. Now, the B cells are very key in producing antibodies to the virus. Usually, at the acute phase, you have the production of immunoglobulin M, which is the first immunoglobulin that is mature from the process of immunoglobulin generation. Then there is a class switch to other forms of immunoglobulins like immunoglobulin G when it sets into chronicity or chronic phase. Then the effectiveness of an antibody response depends on the quality of antibodies, because certain antibodies can either play a role in neutralizing, and others can play a role in opsonization. So it depends on the duration, the quantity of antibodies produced, and the stability of that particular antibody for it to function as required. This slide explains further what I've said, that antibodies bind to viral surface antigens or the PAMs, and this protects against the reinfection by blocking those sites. Huge amount of secretory immunoglobulin A is required, especially to block viral attachment at mucosal surfaces. The viral entry into cells is mediated through receptors, and I've said these receptors can be receptors for many things, inclusive of interferons, the viral epitopes or antigenic determinants, and so forth. So within each immune cell, there are many receptors on the surface to different things as the need might arise. If the receptor is blocked, infection is blocked as well. Like the polio vaccine relies on immunoglobulin A production simply because the poliovirus will try to attack the mucosal surfaces, and this is targeted by the vaccination. Remember, I said earlier on, the role of vaccination in boosting immune response is also to make sure that there's enough generation of either antibodies or memory T cells. This is just an explanation of the same thing I've talked about. I don't want to dwell on this. The key thing here is that there is also the involvement of the T cell mediated immunity, whereby, unlike the earlier vaccines, nowadays people are trying to come up with vaccines that can also stimulate the memory T cells. In addition to you having a vaccine that can generate antibodies, certain pathogens have become a little bit evasive by the fact that there is continuous changing of the antigens on the surface of the pathogens, so that those antibodies will never be active or important in dealing with this infection. So generation of T memory cells is also part of the target by the modern vaccine, so that they can generate T-specific cells that can see that particular antigen and cause lysis or target it for destruction. And these are some of the strategies that are being used by vaccines to produce cytotoxic T cells against the target pathogens. T cell responses, in case of coronavirus. I've just put this one for your general knowledge that yes, certain T cells recognize cells infected with specific virus, for example, the SARS-CoV-2, and rapidly replicate. It has been now known that two different types of T cells are responsible for two functions. The CD8 cells, which are very key in killing the SARS-CoV infected patients, and we also have the CD4, which stimulate the B cells to produce antibodies. Now, evasive mechanisms, various pathogens have evolved certain evasive mechanisms to evade being recognized or being eliminated by the immune system. Like the examples I've just given, hepatitis C virus, they evade by inhibiting the action of RNase-dependent protein kinase. So that one is eliminated, which means the virus will continue to replicate. Herpes simplex virus decreases the expression of MHC class 1, and this avoids CTL elimination. Remember, MHC class 1 is important for attaching with an antigen for recognition by cytotoxic T cell or a CD8. So if this MHC class 1 is not expressed, then it becomes an advantage for the virus to continuously replicate without being noticed. The CMV is also very important because it also decreases MHC. Actually, many viral infections, they decrease the expression of MHC class 1, and that's why they are able to bypass recognition by the immune system, and so forth. So others keep changing their antigens. For example, there is influenza virus that changes their antigen by two mechanisms: antigenic drift, which is accumulation of minor genetic mutations over time, and also there is antigenic shift, whereby there is a recombination of genes from different influenza virus strains. Now, as I finish, what do we know about the immune response to SARS-CoV-2? That most patients recover from COVID-19 have antibodies to the virus detectable in blood, but this is not persistent forever. At one time, they will fade off. The most COVID-19 patients develop antibodies about 10 to 15 days after symptoms start. This is around...
Excuse me, Doc. Excuse me. Hello.
Yes.
Sorry for interrupting.
Yes.
We have another class which we are 20 minutes late. So if you could at least summarize so that we don't get so late. Okay, thank you.
Okay. So I think this one then, because of time, you can read that one. I think let me mention briefly about the bacteria, that they are also critical in terms of eliminating by the humoral as well as the cellular immune response. What is critical about the bacteria is the toxins that I mentioned earlier and the mechanism by which this has to be eliminated, and that is involving the immunity between the antibodies and the T cells. I think in recognition that you need to exceed to others, what is remaining is not a lot. I think we are almost done, and I can stop there so that you don't get late for the other lecture as well. So I can share with you this afterwards. I think that is enough for today.
Thank you very much.
Thank you.